been around. I've had a lot of experience in this country, in France, Canada, and USA, in administration, on ships, in the laboratories, and in the classroom. And that gives you a little bit of insight into things. But here, I'm with working with the group, and this is the way we started originally here with Eco and Ali and Manu and Feather. There are Feathers here, Manu's probably not here. But we started out a small group of people about 5 years ago, and working on electron transport activity, and in the laboratory, but at sea also. The group has evolved, and Igor and Natalia came on board, and let me see, we've got Igor and Natalia, and now Vanessa, and Maite Tamies, and Maite Asensio came on, and then left, actually. Feather was in there in the beginning. So we've had a fairly large team, and what I wanted to do was to report what we've done over these years. And it turns out I bit off much more than I could chew. There were 18 papers to digest, and then 36 or more presentations, and it was it was a big job. So I have selected, but not really exactly by design, partially by what I had on the computer at the time. I put together to give you an idea on what we've been doing. Let me see. Let me put the next thing here. The unifying theme behind all of us here is the respiratory electron transport system. Now, I often get in trouble because I know every Spanish kid studies this in his bachillerato. I know that because my own kid studied it in Ria Domes in Catalonia. However, most people really aren't going to remember it. But this is one of the most basic and most widespread enzymatic systems in life on the planet. It's in respiration, it's in photosynthesis, it's in nitrogen fixation, it's in nitrification, it's in sulfate reduction, it's in the reduction of all of the trace metals in the sediments. Where else? It is just so basic that it evolved about 2 billion years ago, long before there was oxygen in the atmosphere. And it may have evolved even as proteins were being evolved. The basic reactions inside these complexes are catalyzed by trace metals, the same trace metals that you can find in the vents today. And there are theories now that suppose that life began before membranes, but using a system like this to make energy. So it's a very ancient system, it's very widespread, it's in everything. And the way we measure it at this first stage here, if we understand the chemistry of the water around us, we can make inferences about the oxygen consumption or about the manganese reduction, or the iron, the use of iron, sulfate, nitrate. We can make inferences about lots of different types of respiration simply by measuring this first complex, and that's what we do. Okay, so that's the unifying theme, the respiratory electron transport system. As I implied here, the way we measure it, we stick in an artificial electron acceptor, it's called INT, produces an organic compound, which is red, as you can see in this picture here. From the results that we get, you can see we can, as I said, talk about oxygen consumption, nitrate reduction, nitrite reduction, nitrous oxide reduction, sulfate reduction, and then all the trace metals. That's just a starter. Since I made up this diagram quite a few years ago, the electron transport system was found in all kinds of other forms of metabolism. And so nitrification and nitrogen fixation can also be studied by simply making electron transport measurements. Okay, our methodology, all right? The enzyme thing puts people off often. They think, oh, a bunch of biochemists. But the reality is, we go to see we use nets to collect zooplankton. We use CTDs and the rosettes to capture the phytoplankton, the archaea, and the microplankton. And we filter those samples, and we get the electron transport measurements on the particulate matter from the filters. From the zooplankton, we work with the cod end, take the zooplankton that are captured there, make cell-free extracts, and then run enzyme assays on that. So even though we're into the basic biochemistry, the basic biochemistry, the enzymology, the chemistry of these reactions, we apply it to the ocean through taking zooplankton samples, through taking microplankton samples. All right. Okay, I wanted to say some things about the scientific basis. There's something. I'll get to that in a minute. This is a philosophy that, in a way, makes our team unique. Other people use enzymes, but they use them as ecological indexes. You get a green color, and you've got something to do with photosynthesis of chlorophyll. You get a blue color, you can infer another physiological proxy, another process. People can use enzymes the way the geochemists use stable isotopes as proxies for all kinds of things: temperature, ancient phosphate concentrations in the sediments and things like that. If you get a correlation between one thing and another, you can often use the first measurement as a proxy. But we don't do that. We use stoichiometry. And that's the key difference between us and most other people that work with enzymes in marine science. But these are the core things. Plankton physiology changes the ocean chemistry. And as I just said, and I showed you in the previous diagram, if you have respiration and photosynthesis, you're changing pH, you're changing CO2, you're changing oxygen. But it's the enzymes in the plankton that are doing it. And that's what I say here. Enzyme reactions control the phytoplankton physiology. And the enzyme kinetics then control the enzyme reactions. And so that's why we get into it sometimes when we're in the laboratory, into straight kinetics and into straight chemistry. Because we need to understand how the enzyme reactions are controlled by the chemistry if we're going to understand the plankton physiology and then make inferences about ocean chemistry. Okay, philosophical basis. This is something you get when you've been around a long time. Let me see. This first thing here. Some of you may have heard in meetings, some of you may have heard from professors, experimentation isn't useful in oceanography. Oceanography advances by going to sea and making field measurements and then writing good papers on what we find out at sea. And that's true to some degree. But if the measurements are bad, if the methodology is shit, then your measurements and your whole cruise is going to be worthless. And I can tell you from my years of experience that many a ship has gone to sea and come back with absolutely nothing because the methodology was bad. And this is particularly true of biology. The navies of the world put lots of money into physical oceanography and a fair amount into chemical oceanography. But they think the biologists aren't capable of using the money properly to develop technology. And that's a reality. And so the technology in biological oceanography has been retarded for the last 40 years or more. And we have to suffer with that, and everybody else in oceanography suffers. So what's the remedy? The remedy is laboratory work. Good experiments in the laboratory, where you calibrate, where you understand your measurements, so that when you go to sea and you make a measurement, it's worth something. Okay? And being published doesn't mean it's worth anything. That's the other thing you should know. The literature, now people who know me know when I talk about metabolic theory of ecology, know that there are at least 20 papers in Nature, Science, PNAS, and other places that are worthless because the original paper shouldn't have gotten published, It got published because the peer review system didn't have the people. They couldn't find the people to review it properly. And this is particularly problem in environmental science. Okay, anyway, but this point here, the reason I said this is based on Alfred Redfield. Alfred Redfield's a god in oceanography, right? But in 1957, the paper was published in 58, he got up and said experimentation isn't useful in oceanography. And that's what he said in his paper. But there was a discussion afterwards with a bunch of very, very wise people. Bassam? Anyway, no, Arnon, who was one of the co-discoverers of the chemical basis of photosynthesis, was in the audience and chided Redfield for his presentation because he said it was folly. Obviously, experimentation is really important. The panel after Redfield's talk, including Redfield himself, came to the conclusion, yes, experimentation is important in oceanography and we should definitely not exclude it. But what happens is people read that paper, they only read the first part of Redfield's paper where he says it's folly, experimentation is folly, and they quote it. And you'll find people's grants turned down in oceanography, not only in the USA, but in Spain. I remember in Barcelona years ago, Marta Estrada complaining about this. And somebody on a panel had read the paper that says experimentation is useless in oceanography. Therefore, they cut out the lab work in ocean sciences. Okay? All right, this team, our team, tries to do lab work, tries to do good experiments in the laboratory that we can use to interpret our field work. And that's what this is all about here. Oceanography and marine biology are important. Oceanography and biochemical oceanography are important. They go together. All right? Second thing here, let me see. Experimentation, well, this is the part of the first part, is marine biochemistry and physiology experimentation are needed to interpret and refine oceanographic measurements. Methods research. Okay, this is another thing. Probably many of you have heard somebody say, oh, methods research, that's pedestrian. That's the tontos. And they say it. I have heard people say this before. People on faculty committees will say, oh, he's just a methods research guy. Well, methods research guys are people like Golgi, who won the Nobel Prize in the turn of the century. People like, if I can remember his name now, the guy that invented PCR for molecular biology. All of molecular biology is based on Mullis. Kary Mullis, in 85, basically invented the whole PCR technique. He got the Nobel Prize for it, all right? If he hadn't done that methodological research, that whole field wouldn't have mushroomed. And who's the other one? Sanger. Frederick Sanger got the Nobel Prize twice. One for developing a method for sequencing proteins, and then about 12 years later got another Nobel Prize for developing a method for sequencing the bases on nucleic acids. So this guy won the Nobel Prize, right? But anyway, that's just 3 cases that I want you to remember. Methods research is vital to science. It's not pedestrian. It's scientific engineering. The mental processes that go into developing a new method are like the processes that people use to develop GPSs, for example, or your mobile radios, or this little thing like that. I mean, the engineers are thinking. The engineers may not be too verbal, but they know their mathematics and their electronics and their physics. And so what I'm trying, the point here, and that's behind our group, is that methods research is really vital, and it really, if we can make an advance in our methodology, we can open up a whole new scientific endeavor. And so that's part of our philosophy here. Modeling is an essential part of scientific endeavor. Now, this is another thing. There are a lot of experimentalists go into the lab, do a nice experiment, publish the results, and then go on to do another one. They don't think in terms of trying to model or trying to fit their findings into the multidimensional, the matrix of knowledge that exists out there. And that's what modeling does. Modeling takes what you find in the lab and puts it in to the larger framework and the larger picture. But when you do that, you go through a mental exercise so that you understand your own work better. And that means your next investigation is at another level. So the point is, modeling is really a part of laboratory investigation, and it's a major part of field investigation. So in part of our work here, we all get involved and some of us do some modeling, and that's sort of an integral part of the experimental work in the laboratory and as the field work at sea. Okay, let me see now here. Now, some questions, topics. This is what we do. One of the things is looking at starvation's impact on zooplankton respiration. Now, the point is, respiration, as well as any physiological process, will sometime be described mathematically. Right now, it can't be. We have no models, no mathematical models for physiological processes. In all of biology, not just oceanography, it's all of biology. And to be able to write an equation for a process, you have to know all the factors that control it. All of them are different palancas, you know, and you need to know what those things are, and then the mathematical description of how that palanca affects your physiological rate. Okay, focusing down just to look at respiration, and one factor, because there's other factors in respiration, age, growth rate, activity level, sex, time of year, it's all kinds of other things. But we're trying to focus on one thing, because it takes a long time to understand just one process. So one of the things we're looking at is starvation, and for a couple of years we've been looking at it on respiration as well as ammonium excretion. We're interested in understanding how these ratios here of the physiological rate to the enzyme reaction, in all of these cases you have, this is oxygen consumption to ETS, this is ammonium production compared to GDH, this is CO2 production compared to IDH, this is nitrate uptake as compared to nitrate reductase. And we're interested in these variability of these ratios in the sea and in organisms in the laboratory, and what controls them and how do they change. Okay, this is the modeling research here. How can we measure these enzyme activities? I didn't put the physiological rates on there, but they could also be there. How can we measure them more efficiently, more rapidly, more inexpensively? Okay, we always are thinking that when you go to sea, you want to compare your data with the physical oceanographers. Pardon me for thinking, but the physical oceanographers, because of the massive investment through the navies of the world, have a data acquisition rate that we can't match. The chemists have a hard time also, but the biologists have a really hard time in matching the data acquisition rate of the physical oceanographers. And one of the things, why we look at the enzymes is we're hoping that by looking at the basic chemistry behind the physiology in biology, we can increase our data acquisition rate so that we can make real-time measurements, or at least quasi-real-time measurements, to be able to match up our data with the physicists. It's really tough to do that, but that's one of the things. And this leads to the methods development that I've spoken of a number of times here. Okay, how does plankton physiology impact oceanography? Because, you know, we talk about the different physiological rates and we need a measurement. Okay, but what does that mean in oceanography? You know, so you measure a respiration rate, then what are you going to do with that? You know, 50 centavos, maybe you can get or a euro, you can get a cup of coffee with that knowledge. No, really, what are you going to do with those rates? Okay, and this is what we're trying to do, is we're using, in this case here, ETS to actually calculate the flux of different elements, namely carbon, but we're starting with carbon. The vertical carbon flux in the ocean can be calculated from respiration. That idea was fought hard in the USA in the 80s and the 90s. It's a losing battle because it's a reality. If you know the respiration profile of the plankton in the ocean, you can calculate the vertical carbon flux from that. What's the stability of the ocean communities in the epipelagic part of the ocean? That's another thing. You can calculate that stability. You can calculate the efficiency with which they retain their nutrients from the respiration. Who does that? Nobody. The reason is that Maya and I published a paper just last January with the idea, with the concept. But this idea of maintaining ocean communities in the upper part of the ocean and being able to gauge, to measure, to quantify how efficient is, say, the Central Pacific ecosystem in maintaining its community as compared to a Southern Indian Ocean or an Antarctic Ocean. You can do it by measuring the respiration profile. Okay? And that's a contribution. But that's using the enzymes, to the physiology, to the ocean, to ocean ecology to make sense out of a measurement at sea. Benthic metabolism. Well, it turns out benthic metabolism all over the world is controlled by the amount of carbon that is coming down from the water column. Without that, in general, there's a little bit of synthesis, chemosynthesis down there, and that is an unknown, actually. But it still will likely be small compared to the biological production due to the carbon raining from the surface ocean. Well, if you know the water column respiration and the ocean productivity at the top, you can calculate the benthic metabolism. And we're developing models to be able to do just that, to be able to predict what the benthic metabolism is, say 1,000 miles south of the Azores or north of Iceland or wherever, in the open ocean, to be able to, knowing the productivity, knowing some water column respiration, okay, we can tell what the benthic respiration is going to be. Being able to predict anoxia and the growing dead zones off the world's rivers. We know from, in the Gulf of Mexico, for example, the Mississippi River, as it comes out every year, develops a dead zone. And that's anoxic water, and is caused by the respiration of the microbes. And that's the kind of thing we can quantify through our enzyme measurements and be able to predict the extent and the timing of this kind of anoxia. The heterotrophy-autotrophy question. Well, I don't know whether you've followed it, but for 15 years now, the ocean community has been wondering, is the ocean, especially the oligotrophic ocean, is it heterotrophic, is it autotrophic? There have been papers in Nature, PNAS, Science. A lot of people have spent enormous amounts of time on it. Teams, I should say that at least 15 or more teams of people have been working on this problem. And it turns out, after all this work, Pete Williams, one of the founders of the methodology that the people are using, came out and said, okay guys, our method is biased. It's giving us some number that is far off from reality. And teams in Vigo, teams in the States, teams in London, or in England, have spent years making measurements of productivity and respiration in the ocean, trying to calculate this thing. And the methodology they were using was flawed. Another example, because that methodology that Pete Williams developed in 1976 was based on as a photometric endpoint of the Winkler analysis, and the amount of money put into that was peanuts. Not his fault. It was usually the funding system's fault. But it just didn't have the methods development. But he did get a method out, and then everybody all jumped on it and tried to answer this question here. And it turns out they couldn't answer it. The methodology was wrong. Okay, all I'm trying to say is that we're developing our methodology, we're investigating our rate processes to address these kinds of questions in the ocean. That's my point for putting up this slide here. How will the climate affect this heterotrophic-autotrophic balance? But not only in the phytoplankton, not only in the open ocean, but in the in the coastal zones, in the littoral zones, where you have microalgae and seagrass. We don't know very much about these two groups of plants, actually, because they're difficult to work with. It's the methodology there needs some help, and starting to work in this area to apply our enzymology to be able to make respiration measurements, nitrate reduction measurements in macroalgae and in seagrasses. Let me see. The enzyme kinetic thing here, developing enzyme kinetics, this is largely Igor's work, but we're working in this to understand the substrate control kinetics for the ammonium excretion to be able to refine our measurements of ammonium excretion in the ocean. This is sort of an aside, and it came about partially accidentally through Ali Herrera's thesis, but we've developed an interest in cultivating marine zooplankton, marine protozoa in the laboratory. And it's a fun kind of thing for those of you who have ever been aquarium aficionados. We've basically learned how to culture mysids, and we're using them as an experimental tool in our research. Okay, now this gets back to the summary of the research, and we'll start here with the secondary production. Let me see if there's something I've missed. The philosophy I've given you. All right, now what I'll try to do is go through some of the results that we have accumulated, and we do work on this little guy here. We have worked on this little guy. It's a freshwater organism, freshwater zooplankton, but it's easy to grow in the laboratory, and when you don't have much money, sometimes you can turn to this and you can get good results. And in this particular paper here that Mai put together with our team here, we tested secondary production models. Now, the zooplankton people had, up to this time, about 6 models of secondary production. The secondary production is really, it's like primary production, we all know carbon going into the phytoplankton. Secondary production is the carbon going into the zooplankton. It's another term for growth in a way. And in order to measure it in the laboratory, you have to measure growth. And but growth is a long, time-consuming process here. If you look at these, we're talking these are days. So you're talking a long time it takes to follow a culture, different species, and to be able to get their growth rates, and then from those you can calculate secondary production rates. Okay, the modelers tried to find ways of calculating the secondary production rate from temperature and from biomass and from species. And so they proposed a number of these methods, and what we did is we tested them in the laboratory simply by growing Daphnia with different food levels, and then using getting our equations here between weight and the growth rate, and calculating. You can actually calculate secondary production down here from these primary measurements, and then we compared them to what the models would predict. And we used about 5 different, as I say, 5 or 6 different, how many do we have? 5 different models here. And we were able to eliminate most of them. We were only able to find one model that could actually, this one model here by Stockwell and Johansson, that was able to predict the secondary production of this little critter. And yet there were all these other methods in the literature. So this is the kind of laboratory experiment, in a way, is an example of what I was saying, why we need laboratory work in oceanography. In this case, it's almost limnology. Well, no, because some of these models are marine models, some of them are freshwater models. But in aquatic sciences, you need this kind of laboratory work, which is hard to get funding for, to be able to improve your methodology. In this case, test models that people would otherwise go out and use. One of these models, the Huntley model, I don't know whether it's put down here or not. Well, anyway, it was called the Huntley model because of Huntley and Sheckley, was it?
Lovely.
Huntley and Lopez, okay. But these guys are at Scripps Institution for Oceanography. And everybody thinks, okay, wow, these people are Scripps people, this model's going to be good. But it turns out it was worthless. It was based only on temperature, actually. And so it fell very, very easily. It just didn't predict the secondary production at all. So this was an investigation that was used to weed out the efficient from the inefficient models in the literature. Okay, now this is another theme here, and I've got Eco on here because she's the senior author on this particular paper. But I should tell you, that the modelers all over the world for the last 100 years have been using biomass as a proxy for every biological rate that exists. Photosynthesis, for respiration, for ammonium excretion. They'll take biomass and a factor. Biomass and a factor, and then they'll calculate some process. Wow. I mean, a physicist, first of all, or a chemist anyway, who understands the difference between static and dynamic measurements, can note intuitively, actually, if you start taking a static measurement and you try to calculate from it a dynamic measurement, you're starting off on the wrong foot in the very first place. And this is what people do all the time, the modelers. They're just taking a static property, multiplying it by a factor to calculate a dynamic property. Well, in certain situations it works, but certainly not universally, and certainly not to the degree of accuracy that the modelers are now being called upon to work at for climate change. For climate change, we need good models. For ecosystems models, we need good ones. And you can't build you can't predict a dynamic variable from a static one. And we've been trying to argue this point, but the biological community is incredibly resistant to this kind of thinking. Our point is that potential respiration, you can read electron transport in this category here, because electron transport activity is potential respiration, is a better respiratory predictor than biomass. We just focused on Artemia salina, just one organism to show it. But in this case, in this particular paper, we were able to show a really good relationship that was better than the biomass, and we're able to show that this relationship. Let me see. We've got the relationship between the respiration and the potential respiration. There's a lot of fuzz in here, but it holds up. It's sort of steady over time in a culture of these Artemia salina, little brine shrimp. But this work actually really, in some ways, didn't need to be done because it was known. This is a work of a guy named Finlay in England. But he took some work that I did years ago with Randy King on zooplankton, and some work I did with John Christensen on bacteria. And then he did it on a protozoan. What he did was he measured the electron transport activity, measured the respiration, and over, like, I can't remember how many we've got, like 9 or 10 orders of magnitude, you get this linear relationship. You can do the same thing with biomass, it's true. But what happens is that the error range is smaller when you work with ETS. But the point is, this is revealing a fundamental truth about what respiration is. And anybody who takes biochemistry in the university actually knows that the basis of respiration is the respiratory electron transport activity. But the whole ecological community, whether it's on terrestrial or aquatic, somehow bypass. They have a disconnect between what they learn in ecology and what they learn in the school about biochemistry, and they forget that the basis of respiration are these respiratory complexes in the electron transport system, which I showed you in the third slide before, and if you look at a relationship between mass, for example, and respiration, and you start with living organisms, the relationship goes all the way down to the respiratory complexes. It goes through the mitochondria. So Maya and I published a paper a number of years ago in which we pointed out and we said, okay, look, the well-known relationship between biomass and respiration exists solely because the biomass packages mitochondria. And the mitochondria package the electron transport system. And I mean, these are the mitochondria here. These are the respiratory electron transport complexes here. This is a scale of mass. Here's an elephant up here, a mouse. I don't know. The organisms are lined along here. This line up in this part is what's called Kleiber's law. And it started with Rubner's scaling laws 130 years ago. I mean, most of us in our biology have realized, oh yeah, if you have bigger organisms, they're going to respire more. I mean, they're little ones, they're going to respire less. I mean, everybody knows there's a correlation between biomass size and respiration. Well, that's called Kleiber's law in these days now. Okay, and that's what you've got right here. You've got Kleiber's law here. But if you extrapolate it down, it gets to the mitochondria. And then further down, it's electron transport complexes. And so this is what we've been saying, and we said it in Eco's paper, and we said it with mine, is that the reason that Kleiber's law works is because the mass packages mitochondria, and mitochondria package the ETS. And this data here was not ours. This was by a guy named West, who published this in the Proceedings of the National Academy of Sciences in 2002. And he didn't really, this is amazing, he didn't realize what he actually published at the time. He was publishing, in this paper, they were trying to support their theory of the metabolic theory of ecology, which is based on Kleiber's law. And actually they showed the relationship in this kind of thing. They showed the real fundamental basis of respiration in this diagram, mitochondria and electron transport. It was their data, not ours. But so actually, if people had read the literature correctly and understood what they saw, we wouldn't have had to do our star work. There's another case like this, which we'll mention later on. People often don't understand, they don't interpret what's actually in the literature. And even the authors themselves, in this particular case. Okay, so that's Eco's paper. And now I'm going to get on to something that Ali did. Now, I mentioned that we like to culture. We're learning how to culture. We're trying to extend our culturing from, starting with this mycet up here. We would like to get into other organisms. But the first step was represented in this particular paper by Alicia. And what she showed was simply that if you're very careful and you do things right, you can get this mycet to grow in the laboratory up to have a very high survival rate up to 20 days in culture. And we now can get generations and generations. We can raise them from babies in the laboratory. And the nice thing about this is this type of culturing produces mycets which are nutritionally as rich as the mycets out in the wild. Now, this is an extension of our kind of research, gets into the aquaculture because the people who are growing seahorses or octopus or other kinds of organisms can use this organism in their aquaculture business actually to produce very healthy organisms. And so it's becoming we learned to culture this because we wanted to do scientific investigations, but the offspring of it is that it can be used in aquaculture as a food for fish culture. Okay, now starvation. I mentioned we have the starvation. Here's an example of what happens during starvation. You get a decrease. Actually, without my glasses, I'm troubling. This is the respiration here. This is the respiration to ETS ratio. They drop off here. And this is the impact of the starvation on a physiological process. Over here, what you see is the impact of the starvation on a biochemical process. And it turns out the electron transport activity is relatively stable even while the organisms are starving. And Ali published this paper under the effect of starvation and feeding on respiratory metabolism in leptomyces, and that was it. That was it. Now, Igor, working with the same mycid, but working with GDH and working with ammonium excretion, found a similar curve with the physiology and a similar line with the biochemistry. It's simply showing that the biochemistry is relatively stable, largely because the enzymes we work with are what are called constitutive enzymes. They don't change too much due to environmental conditions. So that was Igor's. Now, in doing Igor's work, he had to do some methods work, because the GDH, the glutamate dehydrogenase assay that he wanted to work with, hadn't been, well, it hadn't been looked at for at least 10 years, and we felt that we could improve the methodology by using fluorescence. Fluorescence is a much more sensitive technique than using spectrophotometry in chemistry. So what Igor did was adapt fluorescence and measure glutamate dehydrogenase fluorometrically in the laboratory. This is his, let me see, I've got his, this is like a standard curve. Shows that the GDH activity is linear with the protein biomass, which in effect is actually the amount of enzyme. This is a proxy here for the amount of enzyme here. Then he compared his new method with the standard spectrophotometric method and found a good relationship. Basically, it was a stable relationship. And using that, he got the previous, that's how he got these results here. Let me see if I go back. Okay. In this particular case here, he got these results here. Now this is, okay, I've said we measure the enzymes so that we can measure physiological processes or use them as a proxy for measuring a physiological process. And this is what Igor has done here. He's measured an enzyme activity, glutamate dehydrogenase. This is also in the mitochondria, by the way. And he's using it to calculate in zooplankton the rate at which the zooplankton excrete ammonium into the water. So here we are measuring GDH to give a measure to be able to detect the rate at which seawater ammonium chemistry is changing. So here we're talking about the enzyme chemistry here on this side, and this is an effect on ocean chemistry, because this ammonia goes right into the seawater. Now, what does it do when it goes in the seawater? Well, usually, any phytoplankton around there takes it up immediately and uses it to build protein. So it goes right, cycles back very fast. But that's one of the things Igor is studying. And he took this method with him on the Malaspina program and measured GDH and ammonium excretion on the Malaspina program. Now let me see if I've got this. Okay, and this is real. This is what happens in the laboratory, and this can lead to people like Redfield saying, "Oh, the laboratory work isn't good." But in the laboratory, when you work with a single species under certain defined conditions, you get nice results. You take the same method to seed, and you get that kind of thing. Okay? Now, does that mean you abandon the work that you did in the laboratory? No. That work in the laboratory helps you at least understand what's going on here. The variability that you've got here is simply due to the fact you've got multiple species, you've got multiple times, multiple locations. This is from North Atlantic, South Atlantic, Indian Ocean data. So you've got geographical variability in there. You've got time of day, season. You've got species changes. You've got starvation levels. You don't know when you take a sample whether the zooplankton are well fed or whether they're starved. We don't have an index for that. Well, we could do gut contact. You could do gut. But in general, we don't. We take our samples blind. And this is the kind of scatter that you get from real field data. But nevertheless, when you start saying that, you don't throw in the towel. You don't give up. You start saying, okay, let's pin down, why is this point so far away from the line? And why is this one so far below? And then you go back to the lab and you try to design experiments to test and see if you can simulate this kind of variability using, well, activity or using different species or whatever, using different factors. Okay, now, I mentioned the Malaspina cruise. Nat, Igor, and Feder all participated in the Malaspina cruise. They measured respiration, they measured ammonium excretion, they measured ETS, they measured GDH, they did fractionation. They did a lot of different things in a lot of different places. And because the Malaspina program was designed, well, it's like a big fishing expedition. It was designed to give a lot of geographical coverage, but you couldn't get any time series data. Many times you couldn't go back and check a station, and you just had to collect whatever you can and make the best of it. And this is what we've done. It turns out we're having a hard time actually making good science. This is partially an example of what I said before. You know, you can have a big expedition like this, you can take all kinds of measurements, but whether you can get a good scientific paper out of it is another story. And we're a bit challenged, you might say, in trying to do that. But because of this kind, let me see, this kind of a curve actually will be useful in the way I'm presenting it now. It shows you the reality of field measurements, and that, if we juxtapose that with laboratory measurements and other experiments, will make sense. Okay, this is, now, somebody correct me, let me see. Part of this data, this data here is, see, because Igor didn't put exactly in there, but this is, part of it is the Malaspina, but I also think some of it's Namibia. And it's ETS data, it's GDH data, it's biomass data. It shows just a general trend with the biomass. Look at these are, this is logarithm, so we're going over 3 or 4 orders of magnitude. It's a fairly large, and this is general trend one. But the scatter is enormous, and so that's part of what we're going to have to do is to find out what's causing all this scatter. But that's real field data of enzymes and biomass from the open ocean. Now, as I said, the electron transport system is one of our basic themes. Because of this kind of scatter, we start to look at the controlling chemistry of the electron transport system and see if we can find another way of actually predicting respiration from this kind of data here. Because classically, what people would do with this kind of data, with ETS or GDH data, as I said early in the talk, is they will multiply it by a constant, just the way they would with biomass, to get respiration in this case, or ammonium excretion. And we're thinking that, okay, if you've got starved organisms or organisms that are active in some ways, they're getting more substrates into the electron transport system, or any enzyme, if the GDH case, in the GDH case, they're getting more substrates. And that substrate, those substrate levels serve as throttles, palancas, moving the activity of the enzymes up and down and changing the physiological rates because of that. And so Nat Osma has started to look at the levels of pyridonucleotides in plankton to see, one, if she could measure them, because they were never measured before. There were no papers in the ocean literature on pyridonucleotides. And I should divert a little bit here, but pyridonucleotides are like ATP. They're a major currency molecule. Most of you know ATP is our major currency molecule. But the other one are these things called NADH and NADPH. And they're produced constantly in metabolism. They're energy rich. They're used by the electron transport system to create the EMF and the pH gradient across the inner membrane of the mitochondria, which then drives the motor. It's literally a motor that makes the ATP. But you have to get these things, they have to be produced from metabolism. And that's starting to measure these things. And they're very difficult. Because they're an oxidized, one state is oxidized, the other state is reduced. And it's difficult to separate the two chemically. And that's, well, what she does is now she measures the total amount of the purity nucleotide. And she's found some interesting relationships in doing it. This is her work, and I think it's from, well, it's marine zooplankton. Some of it's from Malaspina, some of it's from the Canary Islands around here. But in 3 different size fractions, she's found that whether they're well-fed or well-starved, or starved, that the NAD, this is NADH, the NADH level is usually higher than the NADPH level. NADPH is usually used in growth. It's the purity nucleotide that most of the reactions involved in protein synthesis, nucleotide synthesis, use this NADPH. The degradative reactions, when you break down glucose, fats, and proteins, you produce this particular compound here. And it turns out there's, in general, much more of this is called the catabolic energy currency than there is of the anabolic energy currency. Now, so you see, compared the blue to the red, the blue is always higher in all these cases. But the interesting thing here is that for the larger zooplankton here, it doesn't really matter whether they're well-fed or whether they're starved. The levels don't change too much over 24 hours. When you get to the little guys, the little zooplanktons, they seem to be challenged, and it makes sense when they're small, their metabolism per mass is higher. And after 24 hours, you can see, at least in the NADH, you can see a drop in their purity nucleotide levels. These measurements, we're just literally, we're in the pioneering phase. If you look at science, one of the very first steps is you make some measurements on something to see how the distribution in time and space works out. I mean, that's the first step in science, is what's out there. And actually, in this particular field of the purity nucleotides, that's where we are, because nobody else has measured it in the ocean. So these are firsts on the part of getting too late. Yeah, okay. Anyway, so this is by Nat, working with purity nucleotides, and it's a first. And she's found, let me see now, she's got, over time, she's found, let me see, this is in Oxyrus, she's found the totals of NADH and NADPH dropping over time during starvation. So this is what happens in starvation, is that, one of our themes is looking at the purity nucleotides drop in starvation. But she's also found this rough correlation between the total purity nucleotides and respiration. And because of this now, we're thinking to replace the way to calculate respiration using enzyme kinetics by measuring these substrates. Now, this S1 and this S2, this is, for the ETS, is NADH. S1 is the NADH here, and S2 is the NADPH. Okay? And we've been working on actually modeling respiration from the either predicted measurements of these things and the ETS measurements and kinetic constants, which we have to determine in the laboratory. Now I'm going to move fast here. In a bacteria culture, if you find that the measured respiration is like that, there is no method in microbiology or in any other form of biology, whether it's in any country, that can predict this kind of a curve, that's respiration over time in a bacteria culture. There's nobody that can predict that from any existing measurements or any existing theory that is widespread in the literature. Okay? But we can, and we do, with our enzyme kinetic model. Let me go back. With this model here, we can predict the respiration like that. And that's what we're trying to do. We're trying to develop this theory so that we can do this kind of thing in a bacteria culture. We're trying to do it in the open ocean. We're trying to learn enough about the pyridonucleotides or the glutamine or the substrates that the enzyme use to be able to make models of a physiological process which can accurately predict the rate. Okay, I'm going to move on here. Borja, this is an example of... Borja's not really in our team. He's a physical oceanographer. But Borja was in my class when a couple of years ago and volunteered to do a model, to take all the bacteria data that we had, put it in a model, and we got this paper out of it. But in a nutshell, I'll try to tell you what Borja did. The results that I just showed you were examples of what Borja did. But this is right here. Borja took the bacteria data. The respiration signal was like that. Let me see now. Okay, this is your ETS signal here. This is your protein signal. This is the same data, the same culture, the same experiment. All right? This is your protein signal. This is your ETS signal. This is time. So we're looking at the changes in a bacteria culture with time. Okay? And what Borja did using MATLAB, he took the model that I showed you earlier, the kinetic model, and was able to be able to predict the respiration very nicely using this as primary input, using the ETS data and using the kinetic model based on substrates and based on enzyme kinetics. And he compared it to the reigning model in the literature, the metabolic theory of ecology, which would take this data here that I'm pointing out here, take that data, and it could only predict this curve for respiration. The real curve was this one, the same as, it's the same thing. Well, maybe what have I got? The scale's different. The scale's got to be different because it's the same thing. But the point is, with the metabolic theory of ecology based on biomass, you cannot predict respiration over a time sequence like that. But with the enzyme kinetic model, you can. Okay. Igor is trying to do the same kind of thing with the GDH. And this is it. He's trying to this is the model that he's going to work on using, this will be ammonium production by the zooplankton, this will be GDH, and these will be the substrates of the GDH enzyme. And this is future work. This is what Igor is planning to do in his thesis. Feder is working with the chemistry of the electron transport system. This particular this is the basic reaction of the of the ETS assay that we use. Feder's working on it. This is what you see in the laboratory when you make ETS measurements. Feder has shown, well, this gets to be complicated, but I'll just say, because the peer review system isn't working or works patchily, you get papers in the literature that shouldn't be there, and then somebody has to make an effort to correct a situation. If you've got a paper in the literature that's telling people to do something and it's false, somebody has to correct the situation. Well, Feder wrote a paper in which he corrected a situation in which there was a guy in Norway that was using the tetrazolium reduction the way we do, but he wasn't adding substrates, and he was getting numbers like this. He was getting levels of activity like this, but because he was working with such biomass, he thought he was predicting respiration in zooplankton, but he wasn't, because the respiration was along this line here, and he was getting things like this. And anyway, Feder investigated this particular reaction that I showed you before here, this reaction, and he showed that it's very sensitive to all kinds of organic compounds in the cell, and that he got these reaction rates different for different concentrations. Whoop, there should have been another one someplace. Well, yeah, I guess that's it. And he showed that you have to be very careful when you do an electron transport assay. You have to do it the right way, the way an enzyme chemist would do it, if you're going to get good results. But it took him a year and longer in the laboratory to do it, because there are a lot of people outside that just couldn't understand the logic behind, or they couldn't understand the theory of enzymology. So Feder had to do an experiment and show people in black and white that the substrate-based assay is totally different from this, what they call a non-substrate in vivo assay, which well, it wasn't true, which doesn't measure anything. It measures reducing compounds in a cell. I'm going too fast to make that really clear to you. It's just that probably in all of your fields, you'll from time to time see a paper that gets in there that shouldn't be there, and it can lead people down the wrong way if it's not corrected. And Fetter did the dirty work in a way of actually correcting the problem in the literature. Okay, this was going back to sea. We went to Namibia. These are results off Namibia. ETS, GDH. This gets into the carbon flux, and this is a whole other story. The whole thing of carbon flux is a topic in itself. As a matter of fact, I gave a seminar 2 years ago on it. Nat's picked this up, and she's shown here how basically you take ETS measurements and you calculate vertical carbon flux. But she went a step further, and working with people in Germany, she compared the carbon flux measurements the way we make them from electron transport measurements to actual sediment trap measurements. And the sediment trap measurements are here. Our measurements of the same flux, we get a different number. So the numbers are different. They're not going to be right on. But this opened the door to future collaboration with the Germans from Wernemünde, and we will be working with them probably in the future to calculate carbon flux using the ETS method and comparing it to the sediment trap carbon flux measurements. Okay, her work resulted in this paper here. And now we're extending it to nitrogen. We're going to calculate nitrogen flux with GDH activities, and Igor's doing this. Let me see. We go on, and this goes on. This goes, and then we get into another whole world of Kleiber, of Kleiber's law, and we won't be able to go into all of that here. This gets into Maite Tamier's thesis here, where she's going to try to use knowledge of the respiratory electron transport system in nitrifiers to be able to predict the ammonium oxidation rate, or the rate of nitrification, which people would like to calculate, to predict now, because it's becoming clear that nitrification is another form of ocean productivity. We always think of photosynthesis as being the force or the process that makes carbon to the ocean. But now we realize that nitrification in the dark waters of the ocean can be producing carbon. And everybody's ignored it, partially because we don't have any way to measure it. Well, Maite's working on a method, on a way to measure this, but using our knowledge of the electron transport system to devise a new sensitive method to measure nitrification in the deep sea. With that, I'll close. This was a model. This was from Maite's thesis, and she's found, well, in working with nitrification, she's realized that we need to update our conceptual model of nitrification in different types of the ocean. So I realized I had too much there. And I didn't have to get Vanessa's work here. Vanessa has got some really nice experiments on the RQ, the relationship between the CO2 production of respiration and the oxygen consumption. It turns out Duarte and Williams and everybody else who works with respiration in the ocean uses a constant. Everybody likes to use a constant. It's so simple. But Vanessa's shown very clearly in her analysis of bacterial respiration experiments that is a variable, and it changes with the age and the physiological state of a culture. So the RQ is not a constant, and that's going to change all of these predictions or studies of the autotrophy versus heterotrophy question in the world ocean. Sorry, Vanessa, about that. But she's got a nice piece of work going. Okay, and I didn't get into Maite Asencio's work on the macroalgae. But there's so much, it turns out. It's just, the truth of the matter is, it's an enormous amount of work. But we're all excited and to keep this work going, and we look forward to years as long as we can stay together. There's always I was just making a comment about research teams, because research teams in science are sensitive to human interests, just the way singing groups are. And you all know what happened to ABBA. You know what happened to the Beatles. You know, this is a type of thing that can happen to a research group. So you have to be very delicate when you work with a research group. And you have to realize that everybody's a prima donna, and everybody's a genius. And you need to think really carefully to keep everybody working together in harmony, because you want that harmony. And if you can get harmony in a research group, you can do incredible stuff. And the literature, I'd like to make a study of that. I haven't done that, but there are cases, especially in England, where you have long-term research groups which have produced Nobel Prizes that, you know, through generations. And a study of the dynamics of those groups would be a very interesting subject. Okay, I'll leave it at that. Rather than go on. Any questions?
Yeah. I have a question. I mean, it's a very simple question. I think that you said that the physical oceanographic instrumentation is better developed than biologic oceanographic instrumentation. Yeah. And you said that it is because the Navy has put a lot of money for physical instrumentation development.
That's an oversimplification, but I know from my experience in the USA, because I was funded, the research that I've done over there was partially funded by the US Navy. I had 13 years of solid Navy funding. So I got in with the Navy group, and I saw the dynamics, and I saw how they work. And the research budgets for the physical oceanographers at that time would be up in the $20 million, you know, whereas with the biological oceanographers, if you could get, for a whole group of people, if you could get $1 million out of them, you were lucky. I mean, they just didn't, well, they couldn't. Density of the ocean and the currents of the ocean are vital for the movements of the ships. A jellyfish bloom is not so important, you see. I mean, it goes down to that kind of thing. But there's another factor. And that is, and this is a philosophical one, is that all biological processes require chemical knowledge, and chemistry requires physics. The complexity of biological problems is incredible. And if you go to sociology, you go from biology to sociology, for example, get into the human factor, behavior, it's incredibly complex. And every time you move up this, go up in the scale, the investigations become proportionally or even exponentially more difficult because of all the factors that need to be controlled or considered. And every time you make this jump, it costs more and more and more. So biological research actually is fairly, to do it really right, is expensive, really expensive. The number of variables that you have to consider are enormous. If you go to fish, for example, you've got to consider behavior. And behavior, that's why I've always liked to work with bacteria originally. Working with a single cell is the simplest biological model to work with, and it's much cheaper and easier to work with single cells than it is to work with even zooplankton. So the reason is the nature of the science is more complex, and its relevance to the major funders, mainly the Navy, is less. So it just wasn't the money put into it, and there still isn't.
Yeah.
But that's life. I mean, that's.
That it can be also related to the people involved in physical oceanography and also in biological oceanography. I mean that not much people is linked to physical oceanography, and so we should have, or we need, very good instrumentation with in situ and continuous measurements, not to spend man time or main time for that. And biology, you have a huge number of people working in biology.
Yeah, but
I go to the biology department, and you have, 30 people working in biology. You come to the physics, you have only 3. If we need, and when we go to a ship, you go 20 people working in biology and only 3 in physics, because we don't have more people.
Yeah, but
that you have a lot of people to hire and to work in biology. We don't have. So we have had to develop better instrumentation to avoid.
But I don't think in the
power.
States, I don't think it's exactly that way. I think in the States you have plenty of physical oceanographers.
Not so many
Really?
Than biology. Biology is amazing. And for that,
well, one of the problems with biology. Okay, this is my own pet peeve in biology, is that too many biologists go into the field without understanding the other sciences that they need in order to make significant advances in their research. I sometimes joke about, if somebody likes to pet a kitten, then they'll go into biology, kittens? And it's more than animal husbandry and taking care of little critters in biology. You need to understand the basic chemistry, and you should understand the physics, even the physics and the mathematics. If the carbon flux, one of the problems we have with the carbon flux, people don't understand how we make our carbon flux measurements. Why? Because we use integration. And there aren't many biologists that know how to integrate, or understand what integration is. I remember talking to my colleague in Germany, a really nice guy. He's got a PhD, he's been working for, years. But he could not understand the way we would say, okay, but you take a respiration signal and you integrate it, and I didn't get into that because I went too fast. You integrate a respiration signal and you get the carbon flux signal. I could not understand that. And my feeling is that actually the biologists, the filter for the biological students, in a way, should make sure that they understand basic math, physics, and chemistry before they attack these things. Otherwise, they're just going to be taking care of organisms in the laboratory. But that's another thing. But the point is, I don't. It's not good to think, okay, well, there's 20 biologists. Well, we can use them like Chinese coolies. That's not a good approach. The trouble is biology needs a lot of research in order to be able to get the data acquisition up to the level in which we can get CTD. For example, you take CTD measurements, or even you do Doppler currents. This business is fantastic, and yet there is possibilities to do that in biology. But we need money. The thing is, for example, the enzymes. If you have developed sensors on batfish, or sensors on these torpedoes. I'm trying to think of the gliders, and you get these things moving. You can map out some of these biological variables and create maps of ocean biological dynamics. You could the excretion rate, the respiration rate, photosynthesis rate. You could map it out. Imagine if you could have a map of photosynthesis. Almost at the same speed that you could get a map of temperature, salinity, and density, or nitrate or phosphate or silicate. This can be done, but it takes the kind of people to develop those sensors have to be electrochemists, combination of electrochemists and biochemists to do that, and plus engineers, and nobody's going to put the money into it. You see, for the private sector, there's no profit. And for the military sector, they can't see how that will help them. We used to joke in the States how that will help protect them from Russian submarines or help them find a Russian submarine. That was whereas understanding the density structure in the ocean helped them predict the trajectory of sound waves and sound velocities. And so the sonar, to be able to interpret sonar signals properly, you needed to understand the density structure of the ocean. And that kind of knowledge actually led to the defeat of Nazi Germany in World War II, is because the oceanographers, literally the oceanographers of the U.S. Navy, figured out how to detect German submarines even when they were hiding under thermoclines. And they figured out, but it was the physical oceanographers working with sound. We're learning how the sound the velocity of sound is determined by the density structure of the water column. I mean, that was vital, and the submarine force of Germany was decimated once they figured these things out. Combination of sonar, depth chargers, and this knowledge of physical oceanography defeated the German Navy. So I know the U.S. Navy they will never forget that lesson, and they will always fund the American physical oceanographers forever because of that thing. Biologists can't, they didn't help so much in that war.
Builds this type of sensor, one of them, to measure one of your parameters, he will be rich. And
I'm not so sure.
It will be a change of biologic oceanography. And I don't understand why, for example, the Navy is linked to the physical oceanography, but money from the National Science Foundation, that is you have a lot of money from the
But they won't develop that kind of methods. That's a methods development program that to do that, as I said, I know it requires electrochemists, biochemists, and engineers, electrical engineers working together, and computer people. So you're talking salaries at around 100,000 a year, American dollars, for a couple of years. You're getting in the million level, and they don't put the National Science Foundation does not put that kind of money. And you'd have to find a totally private organization like the Moore Foundation, or like these guys that made their money in Microsoft. They have money for some things, but they've chosen to use it, put it in other areas. But anyway, it hasn't. I'm pretty sure it can be done, but that's just using my intuition. Anyway, we can talk about this. Anybody else?
Gracias.